As part of the Tapas project, Aqua Biotech Group is carrying out an experiment on an integrated multitrophic aquaculture system in Malta. Integrated multitrophic aquaculture, or IMTA, is the cultivation of species from different levels of the food chain. When grown together, these different species use each other's nutrient waste as a source of food. This experiment is investigating whether species of sea cucumber local to Malta can feed on the waste nutrients from fish farms that might otherwise lead to impacts upon the surrounding marine environment.
The sea cucumber species Oloturia poli has been chosen for this experiment because it is a local species common in Maltese waters and because it can adapt very easily to high temperatures in the sea that can be reached during the summer months. The individual specimens of Oloturia poli are kept in specially designed cages that have been placed at 3 different locations around the fish farm and 2 reference sites away from the fish farm. The specimens are going to be sampled to assess if the Oloturia poli is uptaking the nutrients coming from the fish farm, and they're going to be analyzed using stable isotopes techniques.
At the start of the experiment, the biomass of the sea cucumbers in each cage was the same. Their growth rate is being monitored, and a current profiler is being deployed to track nutrient dispersion patterns within the fish farm area. A remotely operated vehicle is also being used to conduct surveys of seagrass, a species that's used in the Mediterranean Sea as an indicator of healthy environments. The information about the nutrient dispersion patterns can be combined with the sea cucumber growth data to assess the feasibility and effectiveness of the IMTA system.
By the end of the project, we hope to have seen a substantial growth in the sea cucumbers and to have collected a large data set of the hydrodynamic activity within the experimental area. During the study, water, sediment, suspended sediments, and sea cucumbers will be sampled and analyzed for isotopic compositions and organic compounds. The isotopic composition will show the flow of carbon and nitrogen isotopes from the fish farm into the sea cucumbers. This will show us if the sea cucumbers are in fact incorporating the derived nutrients into their diet.
For this system to be successful, the cultivation of sea cucumbers must prevent solid waste from fish farms resulting in unwanted impacts on the sea floor. The better the sea cucumbers grow, the more nutrients they'll remove from the environment, and the more economically feasible their cultivation will be.
As part of my PhD study at the University of Stirling, we are investigating the carrying capacity of freshwater and marine aquaculture. We aim to provide tools for European regulatory and licensing frameworks to develop sustainable aquaculture. I am using a predictive modeling approach to estimate the dispersion and deposition of particulate waste from fish waste and uneaten feed within an IMTA setup. Sediment traps were deployed to quantify nutrient deposition to the seafloor surrounding the fish farm, and the information it generates has been used to test the model's predictive capacity. The model outcomes were used to identify the most suitable positions for the sea cucumbers in the IMTA setup. I chose positions that were predicted to have the highest level of nutrient deposition to improve food availability and the likelihood of nutrient uptake by the sea cucumbers. Suspended particulate matter, bending sediment samples, fish mussel and fish feed were among the potential food sources. The stable isotope and fatty acid content of these items were assessed and compared to those in sea cucumber tissue to see if the sea cucumbers were feeding on the farm-derived organic matter. To assess the impacts of the fish farm on the environment, I took water samples, which I analyzed in the labs at the University of Stirling for dissolved nitrogen and suspended solids, and seabed cores and sediment trap samples were analyzed for organic carbon and inorganic nitrogen.
The main objective of the Tapas project is to provide tools to calculate different types of the carrying capacity of freshwater and marine environments, which can place limits on the growth of aquaculture. These can be used both to develop and support the implementation of sustainable policies for the European and worldwide aquaculture sector.